Coming to hematology, or the study of blood. Now, before we start the study of blood, it is very important to understand iron metabolism, because without that, this whole subject won't make sense. Now, iron metabolism. The first thing about iron metabolism is absorption of iron. So iron is absorbed from the upper portion of the small intestine in the ferrous form. In the mucosal cells of the intestine, the iron gets attached to an intracellular iron carrier. And once iron is once again oxidized in the mucosal cells to its ferric form. So iron is absorbed from the upper portion of the small intestine in the ferrous form. In the mucosal cells of the intestine, it gets attached to the intracellular iron carrier and once again oxidized in the mucosal cells to the ferric form. The intracellular iron carrier delivers a variable portion of absorbed iron to plasma, which is carried away to various parts of the body in combination with iron carrier protein, that is called as transferrin. The remainder iron is probably stored in the mucosal ferritin. Now, transfer of iron. Transfer of iron, the part of metabolism of iron with serum iron, iron binding capacity and percent saturation. A certain amount of iron from the intestine absorbed is transferred to the plasma. Transfer of iron. So in the plasma, iron combines with the carrier protein transferrin. The transport of iron throughout the body is accomplished by transferrin. The serum iron level that is measured actually is the amount of iron bound to transferrin. The level of the iron transport protein transferrin is not measured directly. Instead, it is quantified in terms of amount of iron it will bind, a measure called as the total iron binding capacity. In the average normal adults, the amount of iron that serum transferrin can bind is 300 grams per deciliter. But the actual amount of iron bound to transferrin is around 100 grams per deciliter. Call it the reality of life. The serum iron level is thus 100 grams per deciliter. Thus, only approximately one-third of the available iron binding sites that are normally occupied, the serum transferrin is only 33% saturated with iron. This is called as the percent saturation of iron. Now, the serum transferrin receptor. Transferrin delivers its iron to erythroid precursors and other cells of the body as required. Cell surface receptors are present on the cells for this iron transferrin complex. These receptors are called as transferrin receptor. The number of receptors decreases when a person is replete with iron and increases with depletion of iron. Some transferrin receptors are shed into the plasma as erythroid precursors mature. The shed receptors can be detected in plasma and measured as serum transferrin receptor. Now, the concentration of serum transferrin receptor correlates with the rate of erythropoiesis. Now, coming to storage of iron. Iron is stored in the form of serum ferritin. Iron carried in the blood is delivered to various parts according to the requirement, and the remaining iron is stored in the body in combination with iron storage protein, that is apoferritin. Iron combines with apoferritin to form ferritin, which is the intracellular storage form of iron. Iron is stored in ferritin in the ferric form. The storage form of iron is found mainly in the liver parenchyma and the reticuloendothelial cells. Ferritin is mainly an intracellular storage form of iron. Ferritin is found in plasma in small amounts, and the serum ferritin concentration correlates roughly with the amount of iron in stores. So these are all the things which we need to know about the metabolism of iron.